Hey guys, I finally have your assignment ready. But before I did that, talk about it, I wanted to go through this digestion review notes. This is going to go through all of the things that we have done in the last 3 weeks. So if you still haven't turned in your investigations, or if you got things wrong on there that you want to return in and redo, or you didn't understand parts of it, then you can hopefully understand after watching this review. Okay. So to find the review, you're going to go to the week 4. Click on that. And actually, you can just follow through with these notes. But if you wanted to go through without hearing me talk, you can just go to the PowerPoint. Okay. So here's our review. I forgot to put our learning targets in. But our learning targets are going to be just to review what the macromolecules or organic molecules are, what is digestion, and what molecules are important for digestion. Okay. So we have 4 different macromolecules. Macro just means large. So we have proteins. We find those in meats, cheeses, and nuts. Proteins include enzymes, and enzymes we'll talk about on their own, but they speed up chemical reactions. That's their whole job, but they're a type of protein. We have carbohydrates, found in breads, cereals, and pastas, and sugar as well. Fats and lipids are found in oils, meats, and dairy. Nucleic acids are found in anything that was once living. We don't really worry when we talk about digestion about nucleic acids. Mostly we talk about fats, carbohydrates, and proteins. So carbohydrates are composed of carbon, hydrogen, and oxygen. And there's the smallest unit. One sugar is called a monosaccharide. Mono means one. Saccharide means sugar, so one sugar. Some examples would be glucose and fructose. If you remember back to biology, talking about glucose, we break down all of our sugars into glucose or fructose. Fructose we get from fruit. And the main job of carbohydrates is to provide energy for our cells. Disaccharides di means 2, so 2 or more monosaccharides that are joined together. An example would be sucrose, which is table sugar. Polysaccharides, we have many monosaccharides put together in long chains. And this is generally how our food is. And one example of that would be glycogen, which is animal starch. But again, polysaccharides are just all of these, they're just long chains of sugar molecules. So here's an example of some pictures of the structures. So sugars are a circle structure. You don't really have to know too much about it, but glucose is a single. Sucrose, disaccharide, is double, 2. Polysaccharides have many joined together. Lipids and fats. So lipids are made, again, also of carbon, hydrogen, and oxygen, just like our carbohydrates. Fats store energy, help insulate our bodies, keep us warm. Phospholipids make up the cell membranes. So if you remember back to biology, we talked about phospholipids. And then steroids, so cholesterol, sex hormones like testosterone and estrogen, they usually make up parts of the cell membrane, and they help the cell to absorb different nutrients. This is kind of just a very simple picture of what a lipid looks like. So we have triglycerides, which is probably what you've heard of with most fats. So they have 3 tails and attached to a single molecule. Phospholipids have 2 fatty acid tails attached to their phosphate. Steroids are lots of different kinds of shapes. Nucleic acids are made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And again, back to biology, you probably remember, maybe, that nucleic acids are made of nucleotides, which is made of a 5-carbon sugar, a nitrogen base, and a phosphate group. And basically your nucleic acids, I'm not going to talk a lot about them, but They're made of, or they're DNA and RNA. So they're the hereditary materials. That's their main job. Ooh, should have done that. So here's a picture of RNA. It's single-stranded. DNA is double-stranded, if you remember the double helix. There's a different base in RNA. It has uracil instead of thymine. And also they have different sugars. So those are the main differences. RNA is kind of the instructions to make proteins. DNA is the instructions to make an RNA. So they are basically in charge of any hereditary information. Proteins: carbon, hydrogen, oxygen, nitrogen, and sulfur, made of small blocks called amino acids. We have 20 amino acids. Our body can make 12 of them, but we have 8 that we need to get from our diet. Those are called the essential amino acids. So if we don't get those from our diet, then we can have trouble making certain proteins. Proteins perform functions depending on their shapes. We talked about this a lot in the protein investigation at the beginning of the unit. When we change the shape of the protein, it actually can alter the function or make it so that the function doesn't work. Okay, so the way we change that shape is by heating it up, right? We talked about that when we cooked the eggs. It changes the shape of the protein. The protein then can no longer do its job. There are different types of proteins. We have structural, like muscles. We have enzymes, and we're going to talk about those in more detail. We looked at protein structure. We have primary structure, which is just the type of amino acids. Secondary structure, it's either going to be an alpha helix or a beta sheet, and that depends on what amino acids you have. Tertiary structure is kind of how those are all twisted together, alpha and betas. And quaternary is the major large shape of the protein. If we have a mutation somewhere in here or in our DNA that causes the difference in amino acids, it can change the structure as well. In fact, cystic fibrosis, which is a disease that it affects the salt intake, but it causes mucus to build up in the lungs of the person who has it. It's one single amino acid base difference. So that changed the protein enough to cause this disease that's pretty intense. Alright, so enzymes. Enzymes increase the rate of a chemical reaction. Okay, we have digestive enzymes. They break down food. So they're made of proteins. They lower the activation energy, which is just the amount of energy needed to start a reaction. Most reactions need energy to start. So what an enzyme does is they can lower that activation energy, lower the amount of energy needed to begin that process, which speeds it up. They're reaction specific, so we talked about salivary amylase. Salivary amylase is an enzyme that only breaks down starch into sugar. Okay, so a protein wouldn't be able to be broken down by salivary amylase. It can only do a specific reaction. So the way that they work, here's the shape of our protein, our enzyme, and a substrate is, let's say, starch. Okay, so let's say this is salivary amylase. The starch molecule comes in. It fits in like a key in a lock or like a puzzle piece. Okay, so it fits into a certain shape. Once it fits into that shape, usually what happens is this protein changes shape and breaks apart the bonds between these two parts of the molecule, and then it releases it out. And then another one can come in and do the same thing. Okay. So enzymes are essential to digestion. We would not be able to digest food without enzymes. In your last investigation, we learned about carbohydrate digestion. We're going to learn about protein digestion as well. Maybe fats as well, I'm not sure. But what we learned is that carbohydrate digestion starts in the mouth. with enzymes in the saliva called salivary amylase. So salivary amylase is the enzyme. It's in your saliva, okay, and it causes the start of breaking down carbohydrates. Okay, and it breaks it down from polysaccharides into monosaccharides, so from starch to a simple sugar. And how do we know that? Well, from our investigation from those videos I had you watch, there were two tests. They had the iodine test. So normally when we put iodine on starch, it'll turn black when starch is present. If it's not present, it'll stay that iodine brown color. It won't change color at all. And this will be like a purpley black. So what they did was they put starch in one tube without saliva and starch in a tube with saliva, and then they put the iodine in. The tube that did not have the saliva turned black. Well, they first turned black actually right away, but after time went on, the starch was broken down and it started to turn clear again. Okay, so that iodine, the blackness was going away, which shows that the starch was not there any longer. Okay, so they decided then to use the Benedict solution, which turns the different colors when when sugars are present, and they saw that the tube with the saliva changed color after digestion, which shows that there was sugar or simple sugars in there, which shows you that starch then breaks down into simple sugars. So something in saliva breaks down starch into sugar, and we now know that that is salivary amylase. Okay, so that is, hopefully, I hope that that makes a little more sense for you. So take a look back at your investigation, see if you could add anything or change anything and return that in if you want to get a better score. Alright, I'll talk to you soon. Bye guys.